Good afternoon. This is Kevin checking in with you guys. We're just starting, I would say, really probably week three. I know some of you are still working on weeks one and two, which is fine. But anyway, I just wanted to check in, say hello, and give you a couple of updates or just basically some reminders. Again, this class is designed to work at your own pace. There really is only one due date, which is April 17th. Now, um, I do have due dates, as you can see. Uh, I think most of you are starting in or are working on Unit 2 or have just finished up with Unit 2. Again, it has due dates, but they're not strict. You can go past these due dates. The quiz won't close on you. You do have until the last day of the semester, and you have unlimited attempts, and the highest score is recorded. And I also removed the time limits, so you shouldn't see any time limits either. And again, I want to remind you that the questions are from a pool. So if you do take it multiple times, you may get different questions. And they do come from the outline and my slides. This week, Unit 2, we go through cells, tissues, and the integument. So I do want to talk briefly about a few of those things. All of you who have done the Unit 1 quiz did a really nice job. So keep up the good work with that. And generally speaking, the quizzes flow in a similar fashion as you experienced with quiz one. So, anyway, I do want to discuss a few things in Unit 2. I'll keep it brief because, again, we like to keep our videos short and sweet, but yet full of as much info as possible. Um, so, moving forward, as you finish Unit 2 and get into Unit 3, same thing. We have outlines, slides, and a quiz, but also I wanted to remind you that at the end of Module 1, so if we go back and click Module 1, at the bottom is the Module 1 exam. That's the case for all of the modules. You can see we've got 3 items for each unit, making 9, plus 1, giving us 10, again, for the module exam. And it's 100 multiple choice questions from the test bank, and a lot of them, you might recognize from your quizzes. But anyhow, again, unlimited attempts, no time limit on that, highest score kept. So most of your points this semester are going to come from your quizzes and exams. And then, of course, we do have the McGraw-Hill labs. So I want to look at those briefly as well. So let me come over here. McGraw-Hill Connect, see where that takes us. We're going to sign in. So hopefully all of you have the McGraw-Hill. So let's go in there, take a peek. Good. It looks like the first couple assignments have been completed. So I just want to come over here really quickly and see how we're doing on registration. It looks like Tyler, Rosa, Jaden, and Felipe. So one, two, three. So 4 of you have been in here. Tyler, good. Looks like you still need to do one. I'll show you how to do that. Oh no, you did one. I see what's going on. Okay, good, Tyler. So you're good on 1 and 2. Same with Felipe. Excellent work. Now Rosa, Jaden, and the other students. Let me look at my roster. Ba, ba, ba. So Tyler, Rosa, Jaden, Giovanna, and Aya. All right. Make sure that you guys get access to the McGraw-Hill. There's a 2-week free trial. So you have to sign up using for a McGraw-Hill account using your gateway email. Or you could use another email, of course, but that's the one I recommend. So let me go back to the assignments. Oh. And again, it looks like a few of you have already done the um, first one and even started in on the second one. So this is the view that you'll get once you log in and have access. I think most of you have already seen this, well, four of you. But when you're ready to begin, and again, these are unlimited attempts too, 30 points each, just hit begin, and there'll be some info that you're going to want to pay attention to at the beginning. And again, most of these labs are going to look very similar to this one. So let's see. I'm going to move this one. So, if you've read some of the info, again, a receptor is going to be some sort of nerve ending in essence. So, we oftentimes find receptors are either external or internal. If it's an external receptor, it might be bringing in light, so the eyes, bringing in sound with the ears, bringing in smell with the nose, bringing in taste with the mouth, bringing in touch sensations with the skin. Those are all different types of exteroceptors. And then internally, we have receptors really throughout, especially the GI tract, and as well as around muscles and joints. So when we do have some inflammation or we have something going on, it's very easy to detect that because a signal is being sent to the central nervous system via this pathway, the afferent pathway, which involves the sensory neurons. So the sensory and the stimulus is going to get picked up by the receptor, and then the message is going to get delivered along the afferent or sensory pathway. The afferent pathway takes information from the stimulus to the central nervous system. So we have the central nervous system is composed of the brain and spinal cord. Within the spinal cord, we have what's called an interneuron, which is a specialized neuron that we find. Inter means between, by the way. So basically, between the sensory neuron and the motor neuron, we have this inter or between neuron that allows us to elicit a response rather quickly without having to send the message all the way up the ascending sensory tract of the spinal cord that delivers messages all the way to the brain. Now, of course, that information that's coming in from the sensory, let me click this. So, that information that's coming on this sensory pathway that reaches the dorsal root or the back root of the nervous system, that primarily that spinal cord, we're going to see an interneuron that's going to transmit information depending on the intensity of the stimulus. So if we had a really intense stimulus, we're going to need a really quick response. And so I don't need to necessarily involve the brain to get a response, although the brain does get the signal. This interneuron is going to elicit a connection, an immediate connection between the sensory pathway and the motor pathway. And what happens is that motor pathway is going to be where we're going to elicit a response. So this is what we call the effector pathway. So we have the affector or affector coming from the cause or the stimulus, and then we get an effect along the effector or motor pathway. And we always want to affiliate or associate motor with muscles or glands. So typically, the effector that responds is going to be a skeletal muscle, a cardiac muscle, or the heart, or we could get smooth muscle involved. So your digestive tract, or your respiratory tract, or certainly or urinary tract, the cardiovascular system. All of that is, if it's a tube, is smooth muscle. So we want to keep that in mind, that an effector or a responser is going to involve a muscle or a gland, and the gland could be endocrine or exocrine. So we could have hormones released via the endocrine pathway, or we could get something like sweat or oil or saliva or some other enzyme, something like that from the exocrine or exocrine pathway. So again, this first lab gets into the basics of the nervous system and the basics of stimulus and response. So, some of you, if not all of you, have probably been introduced to this type of concept before. If you've taken a psychology class, you know that the stimulus comes in, the brain processes it, and then we respond accordingly. And certainly, some of that is learned behavior. Some of that is certainly just natural. It's in our code, and how we respond to certain stimuli is just innate. It's born within us. So that takes us in a couple weeks. We'll get into the nervous system a little deeper, but we can start to think about what we call kind of this stress sympathetic fight or flight system where stimulus comes in, it's an intense stimulus, we start to sweat, we start to get blood pressure changes, or they go going up, respiration goes up, heart rate goes up, body temperature, sweat, pupils, everything increases when we perceive stress. So, just like with this situation, we have a simple reflex arc involving pain. It looks like a nail or tack is pushed through the skin. That stimulus gets sent to the nervous system because it's an intense stimulus. We need to remove the finger from the object as quickly as possible. So, if you touch something pokey or sharp or hot, you can see how the arm is going to remove. We're going to get a response or an effect from skeletal muscle in this situation. But like I say, in some situations, like with the sympathetic nervous system being kicked in due to some sort of intense stimulus, the bear jumps out at you or whatever. That's that fight or flight or even freeze response. And a lot of those effectors are going to be cardiac muscle and smooth muscle, in addition to skeletal, as well as the glands, right? We get an adrenaline rush. So the adrenal glands are going to get stimulated to release adrenaline. Um, as well as uh, uh, ACTH, and a hormone that helps with inflammation. Because usually, when there's a stressful situation, it usually involves excess fluid and blood to an area, so that could cause inflammation. So, at the same time, we release adrenaline when we're under stress. We also release an anti-inflammatory called cortisol. Anyway, so this particular activity involves reflex responses, simple reflexes. We call them monosynaptic. So one synapse. And in the clinic, we might see something along the lines of checking to see if the bicep contracts, to see if the tricep contracts. So, bicep contraction, front of the arm or anterior arm, is going to cause the elbow to bend. Triceps response, the elbow extends. Patellar reflex, when the doctor taps your knee, your patellar tendon, we get extension of the leg or the knee. Calcaneal, we're going to see some sort of reflex where the toes are pointing down or kind of stand on our tiptoes. And then the plantar reflex is going to be the toes kind of clenching or flexing. So, what you'll do is, again, read the concepts, read the overview, and then before you begin. This is going to give you some of the basics of what you're going to see as you perform the activity. So the first one, you can see we've got a reflex hammer. There are three different possibilities of how we can utilize the hammer. We have a broader uh, uh, side, the broad flat side. We have a pointy side, and then we have the metal tip. So if we're going to use the broad flat side, we might tap on the tendon directly. And again, a tendon connects a muscle to a bone. A ligament attaches a bone to a bone, just kind of FYI. So we use this part number one when we're doing a patellar uh, response, a calcaneal response, or a triceps response. So these middle three, when we perform the activity, we're going to use the broad side. Now, the pointed side, you're going to use that when we need we may have a smaller tendon, we may have a more sensitive area. So we'd place the finger on the tendon, and then the hammer would hit your finger, and then it would radiate through the tendon. So we see that when we do a biceps reflex. And then lastly, the tapered tip, we might use that on the bottom of the feet to see if we get the toes to contract. So once you're ready, you go ahead and you hit continue to the lab simulation. And then it's going to ask you a question for each one at the beginning. Which part of the reflex hammer do we use for the biceps? So we just mentioned we're going to use the pointed rubber side. But let's say you forgot and you clicked the wrong one. Maybe you think it's the broad side. I'm going to click the wrong one just so you can see what happens if you do, because this is low stakes. Oh, it just says I'm incorrect. May want to strike your thumb for this, but it isn't the best choice. So we hit try again. and then we're going to go with pointed rubber side, hit submit, and it says correct. So then we go ahead, click continue, and that is going to enable us to begin the activity. At this point, you're going to just click one time on the hammer. Note I'm not dragging or dropping. I just clicked it, grabbed it, I let go of the mouse. So now if I come in here, I can move it around. And once I'm ready to push the button or to test the reflex, I just hit my mouse, click your button on your mouse. All right, your mouse pad. Notice I've got crosshairs. And notice too, there's a finger there. Because we're using the pointed tip, we're gonna hit the our finger or thumb. So notice though, I have three different possibilities. Oops, I have three, get off of there. Sorry about that. I have three different possible spots in which to perform. So up here's the belly of the muscle. So we usually don't want to be tapping the belly, but let's say you click it, it's just going to tell you nothing happens, right? Then we're gonna come down here. Notice we've got two options, or it looks like three, four, two. There we go. So I come all the way down the forearm. Again, nothing happens. Huh, what's going on here, right? There we go. We found the right one. We got some sort of response. So we click that, use the crosshairs of the biceps tendon, strike it with the hammer, boom, and then we have to record our observations in the lab data. So let's find that. We click lab data. What kind of movement? Remember, we got flexion of the elbow. And then we can go to phase two. So I'll do one more and then you can do the rest on your own. But I do want to show you these, which part we use on the triceps. Remember, the triceps, we're going to use the broad side. So we click that. And again, if you miss it, it just gives you opportunities until you get it correct. So with this one, we click the hammer, move it around, find. Remember, that's the belly of the muscle. So we want to come a little closer down, not on the elbow, but maybe just a little bit above it, tap. And sure enough, we got a little bit of a response. Looked like his arm went out, so we got extension of the elbow. Get lab data, but again, let's say click flexion. Maybe it looked like flexion. Notice it won't let, is that the correct observation? Oh, wait, maybe it was extension. Boom. I click that, I'm good to go. And then next, go to phase three. All right. I'll stop there. You guys can do the last couple on your own, but that's generally how these work. Oh, I do want to do this last one with you because this is a little trickier. So we see the foot, we've got three possibilities to see if we can get we want the toes to flex. So we want to basically kind of make a fist with the feet. So these dots are where you would start. So it's just asking you to grab the hammer. Oh, I gotta answer the question first. Which part? Remember, we're using the metallic tip. Submit, continue. Okay, now I can grab the hammer. So it's going to ask me to put the tip and start it at one of these circles. So if I click it here, nothing happens. So maybe that's not the right one. Let's try there. No, nothing happened. All right, let's try this one over here. Okay, there we go. So that one over here created some sort of plantar flexion of the toes. So then we come down here to lab data, select the movement. There we go, plantar flexion of the toes. All right. And there we go. That's phase six. And again, we didn't do the other ones, but you can see once you finish, you're going to get something that looks like this. And that's it. You don't have to save the PDF. You don't have to do anything else. You're good to go right here. All right. At that point, it's going to record your grade. So that's in essence how all of these McGraw-Hill labs are going to work. How long they take, uh, some are going to be a little longer, a little more tedious, but for the most part, they're anywhere between 10 and 15 minutes. We're going to exit this simulation. So anyway, you have others. Now next week, I'll do, I'll open these other ones, the endocrine thyroid, diffusion osmosis, so you can see those as well. All right, go back in here. All right, excellent. So just a kind of a brief kind of synopsis of what Unit 2 involves. We go a little bit deeper. We get into the cells a little bit. Again, the smallest unit of life. We're looking at growth, development, reproduction. So this goes through some of the basics of the cell and some of the specialized organs and organelles, and a little bit about the behaviors and the actions of these organelles. Gets into a little bit of acids and bases, osmolarity, body fluids, and edema. We also get into diffusion and osmosis, which you're going to see in units. Let me go back here. Lab 3A and 3B, those have to do with diffusion and osmosis. So I know in Unit 2 I talk about it, but Unit 3 or Module 3 lab activities kind of review those with you. What I do for Unit 2 is a hormone activity because this does introduce you to some of the basics of cellular behavior as well as glandular response. All right, and then it gets into the life cycle. I talk a little bit about mitosis as well. Protein synthesis, which involves amino acids being strung together and synthesized and made into proteins. We're going to see transcription occurring um, inside of the nucleus. So basically, transcription is kind of like making a photocopy of the code. And then we send through the messenger RNA, the messenger delivers the code into the cytoplasm. And that's where we can translate the code. All right, so we photocopy it in the nucleus, and then we translate it in the cytoplasm of the cell. Then we start putting together and stringing together amino acids. Those are kind of the basics of protein synthesis. So you'll see that in your slides as well. And again, I want to reiterate: if they're you can use OpenStax for information for this information, but generally, you can use YouTube, you can use whatever means necessary that makes the most sense to you for going deeper on any of these topics. That's totally up to you. How much you do, how deep you go. I just leave it open to each student because everybody has their own intentions. So if your intentions is to learn a little bit more about mutations, by all means, go into YouTube, type in genetic mutations, watch a video. Crash course videos are excellent. For instance, Amoeba Sisters put out good videos. My point is, you can uh, go as deep as you want to go. I just give you it's a golden corral. Here are all the dishes you could pick from. How much you put on your plate and what you put on your plate, that's totally up to you. But here it is. You can take it and run with it. All right, and then we get into tissues a little bit. We go through some of the basic tissue types. We are going to see four tissue types. You're going to see epithelial tissue, which covers and lines. You're also going to see connective tissue, which is usually just deep to epithelium. Connective tissue connects the lining or the covering, the epithelium, with the underlying tissue, which is usually then going to be the muscular tissue. So you're going to have epithelium, connective, muscular, and then underneath all of that, you're going to have, or within, you're going to have nervous tissue. So we see four types of tissues. Epithelium, again, is a covering or a lining, provides protection, provides a connection with the environment outside and inside, so that we get to see the sensory input as well. There's a basement membrane that connects those layers with that connective tissue. So between the epithelium and the connective, you have a little basement membrane. And then we'll look at types of epithelium: simple squamous, simple cuboidal, simple columnar. So notice that term simple is there three times. Simple just means one layer. Okay, one layer. And then the next term, squamous, cuboidal, columnar, those relate to the shape of the cell. So squamous means we've got a flat cell. Cuboidal means we got a cube cell. Columnar means we got like a rectangular or column cell. Pseudostratified means that we have one that looks like stratified or multiples, but it's really one layer of columns. Then we have multiple flat layers, multiple cubes. So we see the word stratified implies that we have more than one layer. So we have stratified squamous, many flat layers, stratified cuboidal, many cube layers, stratified columnar, which is rare, multiple column layers, but we might see that in the respiratory, digestive, and reproductive. All right, transitional changes, thin to thick, thick to thin. So it transitions from its thickness. So we'd see this definitely in some sort of a bladder typically. Yeah. And we have glandular epithelium that usually is going to coat some sort of line your exocrine and endocrine glands. And notice we do have different types of endocrine and exocrine glands. The exocrine gland is going to have a goblet that's going to secrete the product, so whether it's mucus or oil. Endocrine glands don't have ducts or tubules. We may see some of these glands. These are actually exocrine, merocrine, apocrine, holocrine. We might see these in different areas of the body. Apocrine we oftentimes see in the armpit area or the top of the head. All right, then we have connective tissue connecting. Connective tissue involves cells inside of a matrix. So it talks a few of the connective tissue types. So like blood, is cells in plasma. So it's cells in a matrix. So we do consider blood to be a connective tissue. All right, and then there's a little bit of info on different types of membranes. And then we get into muscle tissue. Remember, three types of muscles: skeletal, smooth, and cardiac. Skeletal is muscle tissue attached to your bones. Smooth muscle is all the muscle that's in the tubes inside your body. And then cardiac, of course, this is really to the heart. And then lastly, we have the nervous tissue, which is going to involve certainly the neurons as well as some glial cells. So usually with neurons, we think of or nervous tissue, we think of neurons, axons, and dendrites. They're little wires that deliver information. All right, there's a little bit on repair. Then lastly, I throw in a little bit of skin stuff. So you'll see the epidermis, dermis, and hypodermis. So again, how deep and how much you want to get into, that's totally up to you. But just know that all of this information can be found just pretty much anywhere you want to find it in YouTube or in the Google machine. So I leave that up to you. Let's see. That's really all I had to talk about today. I just wanted to check in. I didn't want to make this video too long. I probably have said too much already, but regardless, I'm going to go ahead and cut it off here. And if you have any questions, go ahead and shoot me an email, and I'll get back to you if I can, or yeah, when I can. Usually it's very quick. So if you send me an email, usually within 24 hours, I get back to you. But for the most part, I think you guys have a pretty good grasp on what you need to do. Have a good week. I'll check in with you again.